It's my pleasure to introduce our next speaker, who is Abraham Palmer. He's professor and vice chair for basic research at the Department of Psychiatry at UCSD in San Diego. He's interested in GWAS in model organism and tries to understand behavior in mice, rats, and humans. The floor is all yours, Abraham.
Terrific. And can you hear me?
Yes, we can hear you.
And see me?
Yes.
Very good. Okay. Well, I'm very happy to be here and in such a terrific session. I'm going to be changing gears a little bit from some of the talks you just heard, but I wanted to start by just putting in context that all of us at this meeting, I think, are primarily concerned with trying to relate variations at the DNA level, shown here, to differences in observable behavior, whether that's in humans or animals. All right, very good. There we go. And we do that because we imagine that those differences in DNA will change the function of individual genes, whether it's their expression or the amino acid sequence of those genes, and that will lead to differences in the way cells behave. In the case of behavior, often neurons behave. And that will lead to differences in the way brain regions and networks of those neurons interact with one another, ultimately giving rise to observable behavior. And the goal of all of these studies, I think, or one of the goals, is initially to make that surprising connection between a difference in the DNA sequence and behavior, and then to fill in these intervening steps. And it's really by filling in those intervening steps that we begin to gain a novel understanding of different aspects of biology. And one of the goals of understanding biology, of course, would be to identify novel therapeutic targets. And so I'm going to break my talk up into 4 relatively short vignettes, the first of which is going to talk about the identification of a gene, CADM2, which, as I'll tell you, has sort of been following me around for the last few years, identified in human GWAS, and then our efforts to model that gene and to see whether or not it has analogous functions in animal models, in a mouse model. Then I'm going to talk about the importance of genetic background, kind of echoing some things that Kristen Bernard said earlier when she was talking about the importance of which iPSC cell you looked at in the earlier session. And that will focus on 2 genes heavily implicated by GWAS, TCF7L2 and CACNA1C. Then I'm going to go on to talk about using a GWAS system in model organisms in mice to both discover and validate genes, and talk to you specifically about a gene AZI2. And then finally, I'll talk about another story, a gene GLO1, where we've really gotten to a deep and novel mechanistic understanding based on studies in animals. So initially, I want to talk about this gene CADM2. And I first became familiar with this gene based on some studies that I and Sandra Sanchez-Roget and others have been involved with 23andMe, where we have essentially paid 23andMe to collect data about a variety of different phenotypes shown here. Basically questions, batteries of questionnaires, initially in 25,000 subjects, and now we have more than 150,000 subjects available for analysis. And one of the things that we measured there was the UPS-P, which is a sort of an impulsivity scale. And when we looked specifically at sensation seeking, and I won't show the data, but one particular question from the sensation seeking subscale drives this, we saw an association with this gene CADM2. All right, so CADM2 may be a gene that we should think about. Subsequent study that was involved a variety of different groups and relied heavily on data from 23andMe as well as other sources, UK Biobank, also identified CADM2. It was actually the most significant association for essentially, would you describe yourself as someone who likes to take risks? All right, so it's a very simple binary question about risk-taking behavior, and CADM2 was the leading gene there. Subsequently, we've been involved in a collaboration led by Danielle Dick with a number of other members listed, some of whom are listed and shown here, to look at externalizing and using genomic structural equation modeling to put together data from a number of different sources, including that risk GWAS that I just mentioned, and various other phenotypes, to try to get at this underlying or latent trait externalizing, which is a well-developed trait in the psychological literature. And actually in earlier twin literatures associated with human genetics. And that allows us, among other things, to do a GWAS for externalizing And you'll see that one of the leading hits in that GWAS for externalizing, again, is this gene CADM2. And in fact, I won't review it for you, but there's a substantial additional literature about CADM2 implicated in a variety of different traits, very, very strongly implicated in a variety of different traits. And so brushing over some complicated questions about whether all of these associations are at the same SNP and what is the functional consequence of those associations, because I don't feel I have time to get into that, I want to instead shift gears and show you how we sort of passed the baton, as Alex mentioned in the earlier session, and really passed it to ourselves. And so this is work that Sandra Sanchez-Roget has done while she was a postdoc in my lab. Many of you know her for her work in human genetics, but in fact, she's also, like me, works in mouse genetics as well. And so in order to follow up on this CADM2 data from humans, we wanted to make a CADM2 mutant allele. And so we were able to get an allele that had been generated through homologous recombination, which flocks the critical exon of CADM2. We then crossed that to a Cree driver line. The details are maybe not of interest to most of you, but that generated a null allele. And what I'm going to tell you about then is our characterization of animals that were either wild type or that carried one copy of the null allele. And significantly, these animals are otherwise completely isogenic. That is, they're on an inbred strain background, C57 Black 6. So there are no other genetic variations between the mutant and the wild type group, which is a big advantage. And they're also, of course, maintained in a very homogeneous environment, another big advantage. And so we worked with Jerome Young, also here at UCSD, who's adapted a version of the Iowa gambling task for mice. And so without going into details about this task, the animal's in an operant box, and they can make various choices in their effort to obtain this delicious strawberry milkshake. Some of those choices are safe, and some of them are relatively more risky, and I won't say more than that. And what we saw, or what Jared's lab saw when he worked with these mice, is that the wild type animals were more risky, whereas the mutant animals that were lacking one copy of CADM2, so had reduced function of CADM2, showed a less risky behavior, and that was extremely robustly significant. Not shown here, they also showed more exploratory behavior, but in many other behavioral domains were quite normal. In fact, there's nothing observably unusual about these mice when you look at them. And so to conclude that little vignette, CADM2 is implicated by GWAS in a variety of traits, centering around perhaps risk-taking behavior being one critical aspect of that. We produce mice with a null allele of CADM2. We certainly think that that null allele is stronger than the endogenous allele that exists in human populations, but that makes it easier to study. And then we modeled risk-taking behavior in mice using this Iowa gambling task. I think it's important to point out here that whereas I don't think it's hopeless, it's certainly harder to model a psychiatric disorder in animals than it is to model a particular trait such as risk-taking behavior in animals. And so an advantage of this design is that the primary GWAS findings were from sort of a RDoc-like trait or an endophenotype, depending on your parlance. And that made it easier to choose an appropriate model, a behavioral model in animals to test. I think that made our job easier. As I've already tried to emphasize, we had a stronger allele on an isogenic background with controlled environmental conditions. All of those things massively improve our power. And it means that in a sample of about 50 or 60 animals, we were able to get these very robust associations with the risk-taking behavior. And the other nice thing about this approach is not only have we maybe validated that CADM2 is likely to be causally related to this associated locus, but we also now have a reagent, these CADM2 mutant mice, which we can use and are using to try to get into the cellular neuroanatomical circuit manner in which CADM2 is important for these sorts of behaviors. So the mouse models really have the promise of elucidating new biology. And I'll get back to that theme later in the talk. So I've introduced now the idea of using an animal model to study a particular gene. But I want to point out that the genetic background, that is the particular inbred strain on which a mutant allele, is a critically important decision. It's hard to know which inbred strain to choose a priori, but it can potentially have a big impact on the phenotype that's observed. And so really we can ask the question, are all mice created equal? And we wanted to explore the impact of choosing different inbred strains on which to express mutant alleles. And so we chose these two genes, TCF7L2 and CACNA1C, so particularly implicated in sort of body weight and diabetes and in a variety of psychiatric traits. And then we generated or obtained animals that were heterozygous for null alleles of each of those two genes. And as I'll show you in the next slide, we crossed those to 30 different inbred strains. And so the breeding scheme here is, I think, elegant. We have a male animal on an inbred strain background, B6. So this animal is genel. Both chromosomes are identical to one another, except that they're carrying a null allele on one chromosome shown here and not on the other. And we can then breed that animal to female animals that represent 30 different inbred lab strains. And that generates F1 animals where necessarily one of the chromosomes will be from the B6 father and one of the chromosomes will be from whichever of these 30 inbred strains they were bred with. And those F1s, therefore, are genetically identical to one another, except that half of them will get the mutant allele and half will get the wild type allele. And that means we can compare the difference in behaviors or other traits between these mutant animals and the wild type animals, working only with litter mates with animals that are genetically identical to one another, except for the presence or absence of the mutation. And so what did we see? What I've got here are a series of plots where I've sorted the animals by the maximal decrease in the mutant animals all the way to the maximal increase in the mutant animals. So for the 30 different backgrounds. And we're looking at different behaviors here. So here's exploratory activity. And you see in the first third or maybe half of the animals, we see that the mutation decreases exploratory activity. But in the remaining approximately half of the animals, there's no effect on exploratory activity. And it doesn't seem to be systematically related to the exploratory activity level in the wild type animals, okay? So demonstrating that depending on which background you happen to express this mutation on, you might either conclude that there was an effect or was not an effect of the gene on the phenotype. Moving to blood glucose levels, obviously relevant to TCF7L2, we see that a proportion of the animals showed a decrease with the mutant allele in blood glucose, and we see one that was trending towards an increase in blood glucose, with most of them unaffected. And finally, looking at acoustic startle response, again, we see several that show a decrease, we see several that show an increase, and we see several that are unaffected. And so really emphasizing the point summarized here in this table that for a variety of different phenotypes that we looked at for two different genes, we see massive, really common, the common observation is that the genetic background can alter, sometimes directionally alter the effect of the mutant allele on the phenotype. Emphasizing, and I think in my conclusions here, I say, emphasizing that interactions or epistasis can change experimental conclusions. Some alleles that we've worked with do seem to have more consistent effects on a given phenotype, but there's no way, I believe, to know a priori which alleles will have consistent versus variable effects on different backgrounds. And there is evidence that this is not just true in mice, but also in humans. And there's been some beautiful work on, for instance, cystic fibrosis alleles, which are modified by genetic backgrounds. So working in a population where all the individuals have cystic fibrosis, it's easy to see that there are modifiers of the impact of that allele on disease severity. So as always, experimental results need to be interpreted with appropriate caution. And I just want to give a plug to the earlier talk by Kristen Bernard, where she pointed out that similar things are true when working in iPSC cells and mutating individual genes. So far, I've talked about genes that were discovered using human GWAS. But in fact, one of the things that's heavily emphasized in my own work is performing GWAS studies in model organisms. And so far, we've done most of this work in mice and rats, but we're now actually experimenting with permiscus and zebrafish as other systems in which we can do GWAS. So here we're going to do gene discovery using techniques very similar to the techniques in human GWAS. And in this particular vignette, I'm going to talk about these mice, which are available from a vendor, Charles River, CFW mice. So these animals are maintained as an outbred population. They've been maintained by the vendor for about 30 years as outbred. So that's maybe 60 to even 120 generations for mice. Every generation, they maintain hundreds of animals, and they avoid breeding siblings to maintain an outbred or a genetically diverse population. The vendor is able to send us non-litter mates, so we don't have very many first-degree relatives, but we certainly have other kinds of relatives, and that's something I won't talk about, but we've had to deal with in statistical ways. These animals are very inexpensive. So I think when we did the study, they were costing us about $5 a mouse. Now I think the price may have increased to $8 a mouse, but they're very, very inexpensive as a reagent, essentially. And they can be genotyped using a sequencing-based approach, and that's because there aren't high-density SNP arrays. And significantly, when we looked at the breakdown of LD, which is critical for the precision of mapping when doing GWAS, in CFW mice, we found that it was superior to any of the other populations that we were able to characterize. And so that means that this is a population that should give relatively precise localization of causal alleles. And so this was a large study done several years ago by former postdocs in my lab, shown here. And what I've done in the upper panel is just to plot many, many GWAS for many different traits to show you that we found lots of stuff. We studied lots of traits. And also to illustrate that we saw a lot of pleiotropy, that is, individual loci that appeared to influence many different traits, sometimes even from different categories of traits. But what I should say, we also looked at EQTLs in multiple brain regions because we were interested in using those to try to explain behavioral associations or behavioral GWAS findings. So here is such a finding, and this is the GWAS for the locomotor response to methamphetamine. So the animals are given methamphetamine, and then we measure essentially how much do they run around. And you see that we have some significant associations. I want to zoom in on this one. And so here we're looking at a locus zoom plot in the lower left panel. And you see the significant association, that is a SNP and many SNPs in LD with it, with the locomotor response to amphetamine. And you see that that same LD block is also an EQTL for one of the genes. I'm not sure if you can see it, but this gene here, AZI2, is influenced by SNPs in this interval as well. And so that leads to an obvious hypothesis that many of you are probably familiar with, that this SNP influences the phenotype because the SNP influences transcript abundance of this gene AZI2, which in turn influences the phenotype. So we wanted to test that, and so we can test that by creating a null allele of AZI2 in another population. And so this is work by Xin Zhou in my lab. I'll have a picture of her on a subsequent slide. And she used CRISPR to delete 7 nucleotides in an exon of AZI2, which is going to lead to nonsense-mediated decay. And you see here we've used real-time PCR to demonstrate that indeed expression of AZI2 is dramatically reduced in animals that carry either a single or 2 copies of this mutant allele. And that allows us to study wild types, individuals that are heterozygous for the mutant allele or homozygous for the mutant allele. So here I'll have to tell you a bit more about the phenotype. What we do with these animals is on day 1 and 2, we administer the vehicle, so just a saline solution to the animals, and we look for 30 minutes at how much they run around. And they run around a little more earlier in the session, and then they calm down. On the 3rd day, we administer methamphetamine, and as expected, that increases locomotor activity. So you see across that 30-minute session, locomotor activity is increasing. Now in this upper panel, I have the animals that were used for the discovery GWAS, that is the animals that first pointed us to AZI2. These animals have been sorted by the lead SNP for that association. And you'll see that when you sort them that way, you can see an obvious difference in the locomotor response to methamphetamine, depending on their genotype at that SNP. So this is not a population where we've introduced a mutation. This is a population that apparently harbors a naturally occurring variant that influences activity. Then in the lower panels, I'm showing you these engineered animals. And now this is on a B6 background, so a different genetic background, wild types, heterozygotes, and mutants. And you see a very similar pattern where on the first 2 days, the genotype doesn't matter, either for the lead SNP in the GWAS or for the engineered allele. But on the 3rd day, the locomotor response to methamphetamine is rather dramatically influenced by mutation of this gene AZI2. So that, in our minds, gives us some confidence that AZI2 may be causally associated with that allele, as I showed in the diagram 2 slides ago. And I want to emphasize a few points about this. First of all, when translating a gene, as I showed you in an earlier story, between humans and animals, one of the challenges is that we can never study precisely the same phenotype in the animal model versus the phenotype that was used to discover the gene in humans. But we can do that in these animal models. And you saw that in the previous slide. We were able to look at exactly the same phenotype in the mutant animal. And that means that it's much easier to reach conclusions about whether or not we're recapitulating the expected phenotype by mutating a gene. And I think that's what I show here. And that really means that we can do something which feels a lot like replication. So keep in mind, replication in humans typically means doing the same or a similar study in a second population. But we can now think about replication in model organisms by doing a direct experimental manipulation, which is obviously a huge advantage. And again, provides us with a reagent that we can use then to try to start understanding the underlying mechanism. I'm not going to talk about it to this audience because I think there's a limited number of people that would be interested. But in fact, my lab has a center to do this sort of work. We're working in rats, not mice, because rats exhibit, in our opinion, more interesting behaviors than mice do. And we're working in a population like those CFW animals. It was actually created in 1984 by mixing 8 inbred rat strains, and they've now been maintained as an outbred population for 92 generations, maintained by Leos Holbert Woods, who's part of the center. And we have many, many affiliated projects. So both the center and then many affiliated projects, which are working in these animals. And they're measuring phenotypes in these animals. They're measuring microbiome, metabolome, epigenome, gene expression, many, many different kinds of traits so that we can stitch together that data. And so we have really a very powerful system in which to do this. There's a website here, and in the chat later, I'd love to talk with people about that. And lastly, to say it's very easy to share that data because, of course, it's animal data. And that means there are none of these restrictions about trying to protect people's confidentiality. The last story I wanted to talk about really illustrates how we can get into new biology by doing these kinds of studies in animals. And this study starts with our identification of a copy number variant that turned out to be prevalent among laboratory and even wild-derived mouse strains. And so it was a copy number variant where some individuals had 1 copy and some had 2 or even 3 copies of about a half megabase region that included 4 genes. One of those genes is GLO1, and that's the gene I'm going to be primarily focusing on. We were interested because when we did this, 2 groups had recently published relatively high-profile papers, one of them saying that more GLO1 made animals appear to be more anxious, and one of them saying that more GLO1 made animals appear to be less anxious. And so we suspected that both of those things couldn't be true, and we realized that probably both of those groups were working with populations that were segregating this copy number variant, and that was driving the expression differences. And in fact, we knew that when we looked at the presence or absence of this copy number variant in genetically diverse populations, there was a massive EQTL. So this is a minus log P of about 10 to the minus 40, a massive EQTL for GLO1 expression associated with that duplication. Furthermore, when we looked at additional data where a number of different inbred strains had been both characterized for their anxiety-like behavior, the amount of time they spent in the center of an open field, which I won't explain, but is widely viewed as a measure of anxious-like behavior in animals. And we plotted that against normalized GLO1 expression in those same inbred strains, we saw a strong relationship where more GLO1 expression appeared to be associated with more anxious behavior in animals. But we didn't know if that was really due to GLO1 in particular, or maybe one of the other genes in that duplication, or maybe just something that was disrupted by that big insult to the genome. And so to get at that, Margaret Dissler, who at the time was a postdoc or a graduate student in my lab, used recombinering to put in about a 250 kb segment of the mouse genome, a back transgene. She used recombinering to kill 2 of the neighboring genes so that only GLO1 would be expressed in animals that took up this back. And to make a long story short, we found that on the B6 background, the more copies of this back that had been incorporated into the animals, the more anxious they seemed to be. And so that confirmed the idea that more GLO1 seemed to be associated with more anxiety-like behavior. And hearkening back to what I told you about the need to look on multiple strain backgrounds, we also did this on FVB, a different inbred strain background, and we saw the same pattern, that more copies of GLO1 were associated with more anxious-like behavior. I had almost hoped that experiment wouldn't succeed because actually there was no good reason at the time to think that GLO1 would influence anxiety-like behavior. GLO1 is an ancient gene. It's in plants and animals. And what it was known to do was metabolize something called methylglyoxal, which is a non-enzymatic side product of glycolysis and some other biochemical reactions. And it's involved in a pathway that reduces that to D-lactate. That happens in every cell in your body. As I said, it happens in plants and animals. It didn't seem to me that it would really be associated with anxiety-like behavior. Be that as it may, we did a lot of studies initially exploring the idea that maybe methylglyoxal was cytotoxic, because at very large doses, it's cytotoxic. But we got around to looking to see whether or not the transgenic animals had more enzymatic activity and less of the substrate of that enzyme, methylglyoxal, and we found that they did. And eventually we gave up on the idea that it was a cytotoxic thing, and we went back to first principles, and we said, if we know that more GLO1 is associated with more anxiety, and we know that more GLO1 is associated with less methylglyoxal, then there's a prediction that MG would be anxiolytic. And we had assumed that MG wasn't commercially available, but it turns out it's quite stable in solution. So we obtained MG and injected it into animals and found that within 10 minutes, so a time course not compatible with cytotoxicity, within 10 minutes, animals became substantially less anxious when administered methylglyoxal. And so that confirmed the idea that methylglyoxal might be the active agent by which GLO1 could influence anxiety. But we didn't know how it could do that. MG is a small, highly reactive molecule. It wasn't obvious how it would mediate an effect like that. But we did know from some additional mouse studies we had done that at higher supra-anxiolytic doses, we started to see locomotor depression. And at very high doses, we saw frank ataxia and even hypothermia. And that kind of a profile is similar to what you see with GABAergic agents. And so based on that behavioral data and the belief that maybe MG was somehow acting at GABA-A receptors, we worked with Lee Plant here to do patch clamp electrophysiology in neurons in culture. And we saw that just like GABA, MG could activate these GABA-A receptors and could be blocked by a specific inhibitor, which blocks both the actions of GABA and MG, demonstrating that MG appears to act directly at GABA-A receptors. I won't show you all the data, but it turns out that MG appears to be a competitive partial agonist at GABA-A receptors. Unlike GABA, it's not packaged in vesicles and released. It seems to diffuse through cellular membranes. Critically, we see that at physiological concentrations of MG are right on the steep part of the dose response function for activating GABA-A receptors. So based on this and some other data, we believe that GABA-A is an endogenous modulator, I'm sorry, that MG is an endogenous modulator of GABA-A receptor function. And then subsequently, we were able to obtain a small molecule inhibitor of GLO1 that blocks the enzymatic function and therefore increases MG concentrations. We saw that that small molecule inhibitor of GLO1 acted just like MG, that is, it had anxiolytic properties in animals. We then thought about anxiety and depression, and based on different data, which I won't describe to you, we hypothesized that maybe methylglyoxal and GLO1 might also influence depression-like behavior in mice. And that's a little bit of a leap because existing GABA-A agonists don't seem to have antidepressant properties, but we were aware of an evolving literature which has grown since then, whereby other GABA-A agents may actually have antidepressant properties, and particularly this allopregnanolone drug, which was recently approved for postpartum depression. And so we sought to see whether or not any of these manipulations, both the pharmacological manipulations with MG and the GLO1 inhibitor, and also the genetic manipulations, either overexpressing GLO1, as I've already shown you, or using another mouse to knock down GLO1 expression, which I'm only now introducing, we saw very consistent effects on these much maligned tests, the tail suspension test and the forced swim tests. So some very old screening tools that have been used to look at antidepressant efficacy. We also saw it with more chronic tests, so tests that are only sensitive to the chronic effects of an index drug fluoxetine, also are sensitive to the effects of a GLO1 inhibitor, PBVG. And in fact, they act more quickly than fluoxetine. So fluoxetine in this paradigm is not active in 5 days, it takes 14 days to become active. But our manipulation of GLO1 was active in a shorter time frame. And then also looking at chronic mild stress, which I won't describe, but a paradigm in which the animals are stressed for several weeks and start to show various behavioral differences, we were able to block or reverse those differences by using two molecularly distinct small molecule inhibitors of GLO1. And also using olfactory bulbectomy, which is yet another model. And then finally, most recently, we've worked with a medicinal chemist here, Seth Cohen, at UCSD, to develop additional small molecule inhibitors that may have more advantageous properties for inhibiting GLO1, and we find them also to show the same kind of efficacy as antidepressants. And so overall, you see here how doing both gene discovery and biological follow-up, all in the context of an animal system, has made it possible for us to gain really substantial novel biological insights into a new system, which has then been useful for potentially addressing an important therapeutic problem. And I believe I'm out of time. I want to thank the numerous collaborators who I've tried to mention as I went through this talk, and I'm now done and ready for questions.
Thank you, Abraham. This was amazing. There's one talk, and I would unmute Monica. So if you would like to ask your question now.
Hi, yes, thank you. Can you hear me?
Yes.
Okay. Well, thanks, Dr. Palmer, for a fascinating talk. I had a very simple question about part one of your presentation, where you presented some of the data on the CADM2 mutants. I noticed that there weren't any double knockout mice, and I wondered if they were lethal, perhaps, or if there was another reason they weren't included.
Yeah, that's a great question. It's not quite lethal, but we see very few viable animals. Most of them die before weaning, and so they're quite sickly, and we didn't feel that behavior from sickly animals like that would be particularly informative. So that's why they weren't included.
Okay, thank you.
And I actually have a question myself. Did you look in like CADM1 as well? Because this was one of our anorexia hits, and I would be personally interested in this.
Chris, I would love to follow up with you about that. We've gotten into the world of CADM and have hooked up with some other people who've done work on CADM1. And as I think you're probably aware, it does have effects on feeding in animals.
Oh, wow. Okay, great.
Yeah, and actually Sandra Sanchez-Roget, who I mentioned during the talk, would be a particularly good person for you to talk to about that.
Okay, great. Thank you.